thank you for the introduction. So it's another talk about polarimetry, and you will see that it's very complementary to OCT with polarimetry. So my work focuses on collagen. So first, I want to introduce you to this major structural protein in mammals. So it's not found within the cells, but outside the cells in the so-called extracellular matrix. That is very important in many tissues. So of course, you know that there is collagen in skin, but it's not the only tissue. Tendons are mainly composed of collagen, so it's a rope of collagen. Bones, there is a mineral part, but there is also an organic part that is composed of collagen. Cornea is almost composed only on collagen. There's collagen also in blood vessels, lungs, so everywhere. So this protein is characterized by triple helical domains that are, so it's a right-handed triple helix composed of 3 left-handed alpha chains. And there may be interruptions in the triple helix so that it forms networks, but there are many collagen types, and most collagen types form fibers. So this triple helix is spontaneously aligned to form fibers that may further form different assemblies. So for example, in skin, there are tangled bundles of fibers, so it's a very disordered media. And in cornea, there are very thin fibers, only 30 nanometer diameter, that are very well aligned into lamellae that are stacked over each other along the depth of the cornea. So it's a so-called multi-scale structure from the angstrom size to typically the millimeter size. It's called also in biology a hierarchical structure. And moreover, it's a chiral multi-scale structure because you have chiral alpha chain, triple helix. The fibers may be also slightly twisted. And also at the scale of the tissue, for example, in cortical bones, you may have concentric layers of collagen fibers with twisted orientations. So what are the biological issues for collagen? First is really to characterize this multi-scale structure because it's not well known in many tissues. Also to understand how is it controlled, what's the relationship between this 3D microstructure and the tissue function, mainly the mechanical stiffness of the tissue. Also, and from a medical point of view, to understand, to characterize collagen remodeling or reorganization or degradation in pathologies. So to do that, we use multiphoton microscopy. So it has already been introduced by Valentina this morning. So just briefly, in usual confocal microscopy, excitation is provided by 1 blue photon, while in multiphoton microscopy, we have 2 red or IR photons. And this process occurs only when the intensity is high enough, so only within the focal volume, so that will result in an intrinsic 3D resolution. So compared to confocal microscopy, we don't need to have a pinhole in front of the detector to select only photons from the focal volume. Here, all the photons come from the focal volume. We can open the pinhole, we collect more photons, and so we go deeper within the tissue. The other reason is that we use near IR excitation, so it's a transparency window of tissues. And of course, well, practically, the resolution is typically the same as confocal microscopy. So this was the main purpose of multiphoton microscopy when it was implemented by Wattweb. But the other very great advantage is that there are many other modes of contrast than fluorescence. So you can have 2 photon excited fluorescence. That is usually that is the most usual multiphoton modality. But you can have also second harmonic microscopy, 3 photon excited fluorescence, third harmonic microscopy, current, anti-Stokes, Raman scattering, and so on. And so that it's a multimodal microscopy. We can combine all these different modalities to have a multicolor image. Moreover, we can use endogenous signals. So for fluorescence, usually it's fluorescent labels, but there are also endogenous intrinsic signals within tissues. So for fluorescence, It's without the cytoplasm of the cells, there are some NADH and FAD, so we can look at cells, and there are also elastic fibers. For second harmonic generation, it's fibrillar collagen, myosin, microtubules, and that's all. And for third harmonic generation, we look at micrometer scale heterogeneity, that is interface, lipid droplets, or biominerals. So it means that we can combine easily all these different processes because they are excited exactly the same way. We just need a dichroic mirror to record two channels in parallel. And we have different images. For example, here it's an rat carotid artery without any staining, and you see the elastic lamellae and the collagen fibers within the adventitia. And here it's second harmonic and third harmonic generation in a marine lung. So it means that it's really now the gold standard for multimodal 3D imaging of thick tissues. So the question now is, well, for fluorescence, it's quite obvious that we have fluorophores in tissues, but what's the origin of these other processes? So specifically for collagen. So at the elementary response is found in the peptide band. So we are looking at proteins. Proteins are polypeptides, so it's a chain of amino acids with peptide bonds within two amino acids. And in the peptide bands, they are delocalized by electrons here, so that it's a dipole. And when we excite for multiphoton microscopy, we need a very high excitation. So we excite non-linearly the dipole, so it can be sensitive to the environment. And here you see there is a non-centrosymmetric environment with an electroacceptor and electroacceptor and electrodonor, so that it will be the electron will spend more time on the electroacceptor time, so that it's not directly the same frequency. You have harmonics, and at the basic harmonics is a second order. The electric field at 2 omega, so it's a second order nonlinear response that is characterized by a hyperpolarizability. So within the focal value, now we have many peptide bands because these peptide bands are very small. So if we have two peptide bands that have opposite directions, the two electric fields that are radiated by the nonlinear dipoles have opposite phases, so that it results in destructive interferences. If they have parallel orientation, parallel polarity, we sum the two electric fields, so we have a doubled electric field, and the intensity is multiplied by 4. So it means that here we have a coherent process, and with coherent summation of all elementary second harmonic responses, we end up with this formula when we have, of course, the square of the excitation intensity, the modulus square of the elementary response, and the square of the number of alike peptides in the same direction. So it means that we need also high density and non-centrosymmetry at this scale, and that's the reason sometimes, it's a reason why second harmonic signals are found in filamentous or fibrillar protein assemblies. Just to mention, that's true in mammals. In plants, it's the same with the sugar and the polysaccharides. So we have second harmonic signal in plants in cellulosis and starch. So in collagen, you have seen the structure. So all the 3 alpha chains within a triple helix and all the triple helices within the fibers have the same polarity. So that we have an efficient current buildup of the signal, and we can see some second harmonic signal in this fibrillar collagen. It's not the case in non-fibrillar collagen. For example, the membrane here, it's a filtration membrane of the kidney in the glomerulus. There is no signal. It's the same also when we have denaturation of this protein. So denaturation of collagen results in gelatin. That's what we do when we boil some meat. And there is no, in that case, there is no second harmonic generation. So it means that we have a structural probe of fibrillar collagen in tissues, but it's complicated because the signal is proportional to the square of the number of aligned peptides. So it's a nonlinear response that is sensitive to the 3D distribution. For example, at the fiber scale, there may be two fibers within the fiber that have opposite polarity, and we have no signal, while still we have some collagen. It's completely different from fluorescence, that is incoherent. In that case, we add the intensities of all harmonophores, and it scales linearly with the number of chromophores, so it's much easier to quantify the signal. So still we can have some phenomenological scoring of the quantity of the collagen just by looking at the percentage of voxels that have a significant signal, and it's highly sensitive. It's highly sensitive because second harmonic is really highly specific to collagen, so there is almost no background noise. So that's the main advantage of this technique, and also no staining, so it's highly reproducible. And also, as it scales as a square of the density, the contrast is very high also, compared to, let's say, OCT or reflectance confocal microscopy. Still, it's not perfectly quantitative, except when we look at isolated fibers. In isolated fibers, we know that we have verified experimentally and theoretically that it scales at, that it scales at the fourth power of the diameter. Another issue with this coherence signal is the coherence length. So you see we have interference between all the nonlinear dipole radiations, and it's not the same in the forward direction and in the backward direction. In the forward direction, if there is not too much dispersion, the coherence length is very long, and we have almost mainly constructive interferences. While in the backward direction, the coherence length practically is very small. So usually we have a very low signal, except if we have, for example, reverse of the polarity on the size of the coherence length. So if we have some very specific distribution of the nonlinear dipoles. So it means that it's complex. An example is for the cornea again. So I remind you the structure with the lamellae. So the lamellae are very thin, 2 micrometers, and it's composed of very small collagen fibers. So from the optical point of view, it's homogeneous. And that's what we see in the backward direction. There is a small coherent length to probe about 100 nanometers within a lamellae. It's homogeneous. But in the forward direction, we probe the focal volume extend over several, at least 2 lamellae. So we have some crossing of direction because they don't have exactly the same direction. And moreover, we have some reversal of polarity within lamellae that result in a striated pattern that shows the direction of the fibers. So it's positive because of this large coherence length. We see the fiber orientation, so the orientation of the lamellae that gives the structure of the cornea. But sometimes so it's not possible to have forward direction. And due to the large coherence length, we have crossing of this lamellae, so it's not clear what to do with this image. So it's better to use backward imaging to have a smaller coherence length. But then we don't have the direction of collagen, and that's really, from a biological point of view, that's the main purpose of imaging, is to see really the direction of a collagen. So that's the reason why you use polarimetry, to look at direction. So I will show polarimetry with linear polarization and then with circular polarization. So first, the setup. So the setup is a laser scanning microscopy technique with galvanometric mirrors. So we have the wave plates just at the back pupil of the objective lens. So objective lens is moved actually to have the third direction when we do to record stacks of this stack of images to have to reconstruct C3 volumes, and we can have detection also in the forward direction. So the first wave plate here, a quarter wave plate, aims at correcting any ellipticity introduced by all the optical components. The second one to switch to circular, and the third one to adjust the linear polarization. So we have three ways of performing experiments, either with circular or quasi-circular polarization to mitigate any effect of the direction of the fibers, or to have linear polarization and to rotate it, or to have circular polarization, and we can do the difference between right-handed circular and left-handed circular, and to observe some circular dichroism in our experiments. So of course it's quite, well, of course we can also have some analysis here. It's not possible in epi detection, but it's possible in transdetection. So it's possible only in thin sample because the polarization is distorted in 6 samples, and also because usually we have some scattering, so it's not a well-defined analysis of the signal. So practically, we do not do that usually, but when it's possible to do that, we have more information, of course. So there may be also a few artifacts, some scanning issue, also due to the bireference that rotates the polarization, some polarization distortion due to strong focusing because there are some actual components. So we know how to, so we know the effect of this, all these artifacts, and that's also the reason why we have a very simple analysis. You will see no Jones matrices, because here we just have a very simple analysis not to be sensitive to all these artifacts. And the main issue with this technique is that it's slow also, but because we have to record, it's a laser scanning technique, and we have to record images with different polarization. But we can use fast polarization switching with an electro-optical modulator instead of these wave plates. And in that case, we synchronize to line scanning so that it's possible to do some fast PSH. So practically here, you see the polarization changing, and we acquire a stack of images with different polarization. And in every pixel, we can plot such a polarimetric diagram. So the direction of the fiber here is given by the direction of the polarimetric diagram. We know that because we can perform a tensorial analysis. We assume that the collagen fiber have a cylindrical symmetry, that the Klein symmetry applies, and we have only 2 independent tensorial components. And we very easily obtain this equation when we have different parameters. So, of course, average second harmonic signal, this orientation phi, and rho, that is the anisotropy parameter, that is a ratio of the 2 tensorial components, and practically the ratio of the 2 minima. So this is not very practical to handle. We rewrite it to do a Fourier transform, and then we obtain here the mean intensity. The orientation, that is the phase of the Fourier components. So it's coded by this color wheel. So here you see that you have orientation like this, and here it's the opposite orientation. So you have 2 different lamellae. And the black pixel corresponds to pixel where when we have a bad quality of the phase determination that correspond to region when there is no signal. Okay, so I've commented about the orientation, but not about this anisotropy parameter that is obtained as a ratio of the 2 minima, and that gives the ratio of the 2 tensorial components. So we can calculate this, just a change of framework from the peptide band framework to the helical axis. And this parameter is related to the orientation of the peptide band to the helical axis. That is practically the helical pitch. Well, so it's verified, it's okay, it's the right value, but it's the same in all tissues. So when it changes, it's due to, sometimes it blocks, it's due to a disorder, because here it's calculated only for one fiber. If we have different fibers with different orientation, it will modify this parameter, and this parameter will be increased as a function of the disorder. So it's a disorder at the sub-micrometer scale. So we have checked it experimentally in tendon by stretching a tendon under the microscope. And we have seen that when you stretch it, we align the fibers, and this parameter decreases. It can also increase if we have out-of-plane collagen fibers, so if we have collagen fiber not within the imaging plane with out-of-plane fiber. So just to give an example of this, that is not really a biological tissue, still it's a biological tissue. It's on parchment. I think there is. Okay, so parchments where the writing supports in the middle edges. So it's composed of animal skin. So the skin is scratched, it's stretched, it's dried under tension, and then you are left only with, sorry. Sometimes it works very well. So you are left only with collagen fibers that are well stretched within the imaging plane. And one issue with this parchment for conservator scientists is the alteration. For example, due to fire in the libraries or to send to the water by the firemen. So there is an issue with the alteration, and when it's altered, there is denaturation of collagen, so it's gelatin, and there is no second harmonic anymore. But this is too late. What we are interested in is the very, very first stage of alteration, to be able to see it without any sampling of the parchment, and to say to the conservator scientist, be careful, you have to do something with this parchment. And so the first stage are really an increase at the disorder within the fiber. And we have done this in model parchments and also in parchment from the Chartres Library in France. And we see that it increases for small denaturation temperature that are measuring the disorder within collagen. So that's the work of my colleague Gaël Latour. Okay, so we have a very non-invasive way to look at the very early states of degradation in parchment. So what's the take-up message for polarization is that we have in-plane orientation at pixel resolution. We have the disorder parameter within the pixel, and we have also the disorder parameter within the field of view, because as we have the orientation distribution, we can compute, for example, the entropy or the circular variance of this distribution and see that it increases. So here it's a transverse section of cornea, so it's a healthy cornea and a keratoconic cornea, and you see that there is a disorganization of the lamellae. So you may say that here I could do the same just with no polarization resolution, so with gray level images, using image processing. So it's faster, it's less complex, but it's not at pixel resolution because we need a few aligned pixels to look at the direction. And moreover, if it's a dense tissue, there are many crossing of fibers, and it's not easy. So we have done also it in 3D. So here it's a full cornea. So it's half a millimeter thick. And you see the different lamellae with different orientation, and you see it's not the ideal case of only two directions, much more complicated, and that's the main purpose of this work, is really to look at the very detailed structure of the collagen in cornea. So one difficulty here is that there may be artifacts in depth, and so we need to validate the measurements in depth, and we don't know the structure of the tissue and want to do it in the same tissue. It may be different from one sample to another one. So the way we did it is just to image the cornea with this position, then to rotate it by 90 degrees and to check that the orientation were also rotated by 90 degrees. And it was true for the first two-thirds, so about 350 micrometer, and then the last third, not so true, but with symmetric artifacts, so it was possible to average the two orientations. Okay, but we have many information on 2D orientations, that is in the imaging plane. So how to have the third orientation is with circular polarization. So I've explained you that collagen is chiral. So as it's chiral, it has a different interaction with chiral light, that is with circular polarizations. And we can compute the difference between the normalized difference between the images recorded with left-handed circular polarization and then with right-handed circular polarization. So it's not very easy because the signal is very low. So we need to accumulate the signal to improve the signal of a noise ratio, and then there is some mechanical drift. So we have the same issues as in super resolution microscopy, and also the same experimental solutions. And also it requires, of course, perfect circular polarization to not to have a linear dichroism contribution. But then when we do that, we have this type of image. So it's, again, a cornea, but here a transverse section. So we may have, in the imaging plane, we may have lamellae with different orientation. So you see in the green color, the usual second harmonic intensity, and the circular dichroism is in purple color. So it's anti-correlated. It's better seen on this transverse profile. And as we know that usual second harmonic signal is maximized for fibers that are within the imaging plane, it means that here what we see is out-of-plane fibers. So it's very interesting because we have no signal from in-plane fibers and only signal from out-of-plane fibers, while in usual second harmonic microscopy, we have a lot of signal for in-plane fibers and not so much from out-of-plane fibers. So it highlights specifically out-of-plane fibers that are not well visualized using usual second harmonic microscopy. So it's really very efficient, but then we can go further and try to know what is the sign of the CD. So in linear optics, the sign of the circular dichroism is related to what enantiomer we are looking at. But in collagen, we have only one enantiomer. So it's a homochirality of life. There is only one enantiomer for all proteins in biological tissues. So it was proposed that the sign of the circular dichroism was related to the out-of-plane angle. It means we have to do the calculation again. So it's again the calculation. So here you have the two usual, well, we have three here, but it's almost two usual components that are present all the time. And then we have added this chiral component that was neglected in the former calculation. So this chiral component is the one with three different subscripts. And we consider that we have probably real chiral components because we are far away from resonances, but this one may be imaginary because we are really very far away from resonances, completely in the Kleinman approximation, we have no chirality. So when we do the calculation, we obtain that the circular dichroism proportional to the imaginary part of this parameter and to sinus of the out-of-plane angle. So it means that it's zero in the imaging plane and it's positive if it points upwards and negative downwards, or vice versa. So it was proposed in a few papers when we started this work, but it was never experimentally verified. So what we did is we just took a cornea section and we flipped it, and we checked if it was the sign was reversed, and it was not the case. It was the same sign everywhere. So not perfectly the same images because as it's a coherent process, it's very sensitive to the exact position of the pixel, but it was really the same sign, and we did it in many samples. We asked our colleagues in a François Légaré group in RS at Montreal to do the same experiments on marine fascia, and we checked that with a crystal we can reverse the sign, so it was strange. So we thought about the theoretical analysis, and we realized that this theoretical analysis had been done in the electric dipolar approximation, while we know that optical activity in linear optics requires to go beyond this electric dipolar approximation. So it means that here we have to add magnetic dipolar contribution and also electric quadrupolar, but I dropped it. So when we do that, we have new tensors. These two new tensors here. So they have also chiral and achiral components. So it's reversed compared to electric dipolar because we have pseudo-vectors. And also there is a 90-degree phase shift compared to electric dipolar. So if we consider that this one is real, it means that this one is imaginary and all these ones are complex. And then we do again the calculation that is quite long, but we can group the components. So we have always product for chiral multiplied by achiral, real by imaginary, and we retrieve the usual inner electric contribution, but we have also inner magnetic contributions and cross contributions. And what is important is that this cross distribution have an even symmetry versus the out-of-plane angle. So it means that they prevail in collagen. And so we have done some numerical simulation with prevalent cross contribution, and we have obtained that, yes, we can obtain no signal for in-plane fibers and the same sign, whatever is up or down. Still, you see that in this image, we have plus and minus sign. So it means that we need to have plus and minus CD. So we did it, but with different susceptibility tensors. So it seems strange, but not so much. Because when we look within focal volume, we have not only one fiber, we have many fibers. If they have the same polarity here, we just double the achiral and chiral parts. But if they have anti-parallel polarity, so the non-chiral part vanishes, but not the chiral part, because an helix at the same polarity, whatever, at the same helicity, whatever the polarity. So it means that here and here, it's not the same susceptibility tensor. So it means that here, we are sensitive to the polarity distribution of out-of-plane fibers within the focal volume. So we have found up to now no application of this technique. Well, we see that there are different organizations within the cornea, but what's the interest of this application, of this technique, I'm not sure of this, but we are still looking to a relevant application in biology. Well, so what is interesting still is that CD highlights out-of-plane fibers, and it's a disorder parameter, exactly like with linear polarization. That is, with polarimetry, we have disorder parameter within the focal volume. Okay, so I have talked a lot about collagen. We can try to look at other proteins. So to do that, it's experiments with my colleague Pierrat at Loeb. We have looked at a zebrafish embryo, so a unique sample with three different proteins that exhibit second harmonic generation, so collagen, myosin, and microtubules. So the first observation is that when you look at the incoming excitation power, we need less power for collagens than for myosins and for microtubules. It means that the signal of collagen is higher than the one of myosin and the one of microtubule. And it's really this, well, collagen is higher than myosin and much higher than microtubule. So that's the first way to compare these proteins. But it's not very easy to compare the intensity when, for example, we are in depth and there may be some depth attenuation. So it's not a right parameter. It's much better to use polarization, because with polarization, when we calculate the rho parameter, the anisotropy parameter, it's a ratiometric parameter. So it's independent of depth. And here we have also differences between these three proteins. So it's 0.5 for myosin, 1.4 for collagen, 2.5 for microtubules. So it means that using polarization resolved microscopy, we can discriminate different proteins in tissue. For example, here we have a mix of the two proteins, and we see that we don't have the same raw parameter for collagen here and for myosin. We can go further and look at the modelization of this system. So we consider that in the other protein, it's the same elementary response along the peptide bond. But in collagen, it's an helical structure. We have done analytical calculation. It's very easy. While, for example, in tubulin, it's much more complicated. So here, we have to do a numerical calculation that is mainly a framework calculation based on the structure from the protein data bank. And we just add this elementary response from the peptide bonds without any contribution from the amino acid residues and without coupling between the peptide bonds. So it's just a coherent summation weighted by the orientation. And we can do it for different conformation of tubulin. Here you have different conformation depending on if it's bound to GTP, Taxol, that is an anti-cancer drug, and we obtain different intensity and different anisotropy parameter, so that we can distinguish between these different conformations that correspond to different microtubule polymerization capability and that are related to different properties of these cells to differentiate and so on. So it's implied, for example, in many cancer signaling pathways. So it's very interesting. So we can go also further and look at all these proteins. So here it's collagen, myosin, and do again the different calculation, and we obtain consistent results. So it's consistent with what we have done experimentally, and we see that it's a good agreement. But what is sure is that for collagen, we have one angle that is a helical pitch. It's also true for myosin, not so much, but not at all for tubulin. Tubulin, there are many angles, so it's not a flat distribution. If it were flat, it would be centrosymmetric, so no signal, but there is no specific angle. Okay. So it means that we have a third advantage of polarization to discriminate these different proteins. Okay. So just to continue a little bit with polarization, I will present rapidly polarization resolved third harmonic microscopy. So first, I present third harmonic microscopy. So here, the response at the molecular level, there is no symmetry requirement. So everything has a third harmonic response. But in tissue, you have no signal. So why? It's because of it's a process that is highly sensitive to the phase distribution. So when we focus a beam, we have a phase shift that is called the Gouy phase shift from -infinite to +infinite. And within the confocal range here, we have a pi/2 phase shift. So this phase shift adds to the usual phase matching term in a third harmonic generation. We have 3 photons at omega that give pi/2, minus 1 photon at 3 omega. So it means that we have 2 times pi/2, that is pi. So it's a minus sign. And we have destructive interferences between this part and, well, before and after the focus. So it means that we have no signal in homogeneous medium. But if we have heterogeneities, for example, 2 different, we can't see that it's a slightly different color. If we have different medium here, that is optical heterogeneities, we have a third harmonic signal, or another way is to have birefringent media and to have the fundamental harmonic beam on 2 different polarizations. So an example is here. If you have a slab, you have a maximum third harmonic generation for typical 1 micrometer, so it's the different cell interfaces. And here with beads, it's lipid droplets in rat hepatocytes. So that's the main work from Emmanuel Beaurepaire in my lab. So now, what to do with polarization? So we expect that it will be the same as with second harmonic generation, that we will look at main orientations and disorder parameters. So let's look here at human skin. Here, it's what we look at. It's lipidic, some lipids at the surface of the skin. And what we see in these lipids is really we probe the direction of the carbon-carbon bonds. So we have a direction that gives the direction of the carbon-carbon bonds within these lipidic layers. And the contrast may give something about the structural disorder, but there are also here aberrations, so it's not very easy. What we can do also is to look at autolytes. I can't have the... So here you see there are some small minerals and beads within the inner ear of this zebrafish embryo. So it's the autolytes, that's the biominerals that we have in the ear. But the question, the biological issue was to determine it was at this stage of development, it was already crystals or only amorphous phases. So we have added some polystyrene beads within the ear, and we see that the polystyrene bead has a different response as autolytes. So it means that here we have a crystalline material, and here we have an amorphous material. So we can see that it's crystalline due to we have the orientation of these small crystals, and we have the disorder. So it's well ordered here. Another example is with kidney stones. So here we have these kidney stones, and we have a very basic analysis. Only with Fourier transform we look if it's one peak or two peaks, because the response, there are many tensorial components, and moreover, there are many orientations of the crystal. So we need two angles for the crystal orientation and 3 or 4 components for the tensorial response, so that we have a very easy analysis. If it's 2 peaks, we know that it's a birefringent material. If it's 1 peak, it's vertical interface, and for horizontal interface, maybe one on the other. And then, if only with this very basic analysis, we can differentiate 3 different kidney stones that correspond to 3 different pathologies that have to be that needs different drugs. Here you have a monohydrate and dihydrate carbon oxalate, and here carbapatite. And you see that for monohydrate carbon oxalate, it's mainly a birefringence third harmonic signal, while here it's related to surfaces. So here it's vertical surfaces, and here either vertical or horizontal. So these experiments are quite sensitive to optical aberrations, so we have to be very accurate in the measurements. So that's my last slide. So you see that with polarization, with linear polarization, we have main direction and disorder parameter. With circular dichroism, it's the same for out-of-plane fibers. So it means that we have really advanced structural probes of tissue in a label-free and non-invasive manner. So I want to acknowledge all my colleagues at Laboratory for Optics and Biosciences, so in the advanced microscopy and tissue physiology. So we have a lot of people in this group of tenured scientists, and all the people in red are involved in this polarization resolved experiments. So Emmanuel is more for third harmonic generation, and myself more for second harmonic generation. All the PhD students, postdoc and alumni also involved in this experiment. All my different collaborators and grants, and thank you for your attention.